Hi everyone. Today I am going to discuss about biofertilizer. It is also called microbial inoculants. That means biofertilizer produced from bacteria, fungi and algae. And it can be defined as the live microbial formulation. Biofertilizer used to improve soil fertility and also it promotes plant growth. It is a non-hazardous way of fertilization. And this biofertilizers can reduce the cost of fertilizer. Okay. And also it serves the economy of the country. The two most important techniques for the application of biofertilizers are bacterization and green manuring. Bacterization means the seeds are treated with bacterial suspension. That is, seed can be treated with Rhizobium, Azotobacter or Bacillus or any other bacteria or other organism. Okay, that is the bacterization. Green manuring is a farming practice. Next, we can discuss advantages of biofertilizers. Biofertilizers have certain applications or advantages over the chemical fertilizers. Okay, that are given here. It can be used by small and marginal farmers because the production rate is cheaper. There is no chance for pollution. Biofertilizers increase the physicochemical properties of the soil, that is, soil texture, soil structure, water holding capacity, pH, cation exchange capacity, etc. Then the algal biofertilizers increase rice yield. Okay, another advantage. Then cyanobacteria secrete some growth promoting substances like NAA, amino acids, IBA, IAA, proteins, vitamins, etc. This all are help in the growth stimulation. Okay, that is, this substances increase the growth of plant. Then another biofertilizers, that is Azotobacter and Azospirillum, that supply nitrogen to the soil. And also it secretes some antibiotics. That antibiotic act as biopesticides. Azolla supply nitrogen, and the azolla can increase the organic matter and also it increase the fertility level and it show tolerance against heavy metals. Then Mycorrhizae make the host plant available with certain elements. It can reduce the plant response to the soil stress and increase the resistance in plants. It increases the life and surface area of roots. So, the mycorrhizae can increase the yield rate. So, these are the Advantages of the biofertilizers. Biofertilizers include rhizobial biofertilizer, Azotobacter, Azospirillum, Azolla, Cyanobacteria, phosphate solubilizing microorganism, Frankia, then mycorrhizal biofertilizers. First, we can discuss rhizobial biofertilizers. Rhizobium is a gram-negative rod-shaped soil bacteria, and this bacteria can fix the atmospheric nitrogen and make the nitrogen availability to the plants. Rhizobium is able to form nodules with legumes. So, there is a symbiotic association between rhizobium and various leguminous plants. Rhizobium is a flagellated organism and it infect and nodulate specific legume host. So, it is divided into several species based on the legume species they nodulate. Okay, that include Rhizobium leguminosarum, Rhizobium fascioli, Rhizobium trifoli, Rhizobium lupin, Rhizobium meliloti. Okay, Rhizobium leguminosarum infect pea plant. Rhizobium fascioli infect Fasciolus species. Then Rhizobium trifoli, Trifolium species. Rhizobium lupin, Lupins. Then Rhizobium meliloti, Melilotus species. Okay. Rhizobium has plasmid. Plasmid P5YM. And this plasmid coat for nodulation and nitrogen fixation. Bacteria enter through the root hair and progress through several steps, and finally it forms nodule. Inside the nodule, bacteroid formation occurs which produces nitrogenase enzyme. This nitrogenase enzyme helps in the nitrogen fixation. Nitrogenase enzyme is sensitive to the oxygen. So, it is protected from the oxygen by leg hemoglobin. Leg hemoglobin is a pink pigment, and this pigment can bind to the oxygen produced by the legume and present in the nodules. For the cultivation of rhizobial fertilizer, inoculum is prepared by growing in yeast extract mannitol broth or yeast extract mannitol agar. Simply, we can call this media as YAMA media. YAMA stands for yeast extract mannitol agar. Acetobacter as biofertilizer. It was first isolated by Bjornik in 1925. It is a non-symbiotic microbe and it is gram-negative, ovoid or rod-shaped soil bacteria. And it is aerobic in nature and it is a free-living nitrogen-fixing bacterium. Azotobacter is heterotroph and it derives energy by degrading plant residues. It is found in slightly acidic to neutral and alkaline condition. Azotobacter can produce indole acetic acid, gibberellic acid, and vitamins. This all can promote the plant growth. The inoculum preparation for the biofertilizer production is same as that of the rhizobium. That is, inoculum is prepared by using Yama media, yeast extract, mannitol, agar media. Then the combination of soil and farmyard manure in the ratio of 1:1. Okay, that is equal proportion of soil and farmyard manure can be used as the carrier. Okay. Azospirillum as biofertilizer. It was first isolated by Bejernik in 1925. Azospirillum is a gram-negative rod-shaped bacteria and it is motile with long polar flagella. It is a vibroid free-living bacterium. Azospirillum is able to fix atmospheric nitrogen in the root zone, which is called associative nitrogen fixation. Azospirillum found in the surface of the plant root, rhizosphere, and also in the rhizoplane region. Okay. Azospirillum lipoferum and Azospirillum brasiliensis are the generally used biofertilizers. Then the cultivation of Azospirillum. For the cultivation, transfer a loopful of the Azospirillum to Ocon's medium. Ocon's medium contains ammonium chloride. Then incubate this medium at 35 degrees Celsius for 3 days. After 3 days incubation, the cells are harvested from this tank, then inoculate in a fermenter containing Ocon's medium. Okay, this is the cultivation method. Next biofertilizer, Cyanobacteria. Cyanobacteria, also called blue-green algae. These are photosynthetic prokaryotic free-living organism, and it can fix nitrogen asymbiotically. Okay. Nostoc, Anabina, Tolipothrix, Plectonema are the well-known examples for nitrogen fixers. 2 or more genera are used for the field inoculation. It is more better than the single strain inoculation. Okay. In cyanobacteria, the nitrogen fixation occurs in a specialized structure called heterocyst. The genes responsible for the nitrogen fixation are recognized for the fixation to take place. Next, cultivation of cyanobacterial biofertilizer. First, shallow trays of iron sheets or polythene lined pits are prepared. Then 2 to 3 kg soil and 100 gram of superphosphates are added. Then the pit is filled with water. Here the pH adjusted to 7 with lime. Then after the pH adjustment, 100 gram of inoculum sprinkled on water surface. Then it is incubated at 35 to 40 degrees Celsius. Then the cyanobacterial mass is separated from the soil after drying, collected and powdered. Then it is stored in polythene bags. Azolla. Some of the known species of Azolla are Azolla mexicana, Azolla pinnata, Azolla caroliniana, and Azolla microphylla. Azolla, also known as mosquito fern, fairy moss, water fern, or duckweed fern. It is an aquatic fern belonging to the family Azollaceae or pteridophytes. Azolla looks like conventional fern. It floats on the surface of the water by means of numerous small, closely overlapping scale-like leaves. Okay. So, it floats on the surface of the water by scale-like leaves. Okay. Scale-like overlapping leaves. And their roots hanging in the water. It forms a symbiotic relationship with Anabaena azollae. This relationship allows the plant to fix atmospheric nitrogen. Azolla shows tolerance against heavy metals like lead, silver, copper, cadmium, chromium, etc. Next, we can move to the production of Azolla. Production of Azolla is simple, that is, water-filled pots inoculated with Azolla, that is, 0.5 kilogram Azolla required per meter square. Okay. Then these pots are left at 15 to 30 degrees Celsius. Insecticide furadon added to the water. Then allow to stand for 3 weeks. After 3 weeks, Azolla mat harvested and then dried. Then it can be used as the green manure. Next biofertilizer, phosphate solubilizing microorganisms. Example for these phosphate solubilizing microorganisms are Pseudomonas, Bacillus, Penicillium, Aspergillus, etc. This organism can be mixed with farmyard manure and can be applied. And it can convert insoluble phosphate into soluble usable form of phosphate. Okay, so insoluble phosphate converted to soluble phosphate by using these organisms. Okay, so it is a good biofertilizer. Next subtopic, Frankia induced nodulation. Actinorhizal hosts are non-leguminous perennial plants belonging to 8 angiosperm families. They are capable of forming root nodules as a result of infection by nitrogen-fixing actinomycete. It is called Frankia. Example, Alnus, Hippophae, Casuarina, Gymnostoma. Frankia is a filamentous branching gram-positive actinomycete. It is present in the soil, but it is difficult to isolate. So, most originate from root nodules. A selective media, that is BAP media, used for the isolation, and it contains potassium hydrogen phosphate, magnesium sulfate, calcium chloride, biotin, iron sulfate, PDTM, etc. Antifungal agents are added to the media to minimize fungal contamination. Frankia are heterotrophic aerobe. We can move to the final topic, that is mycorrhizal biofertilizer. We have already discussed mycorrhizae, types, functions, etc. Mycorrhizae is the mutual relationship between plant root and some fungi. There are three types of mycorrhizae: ectomycorrhizae, entomycorrhizae, and ectendomycorrhizae. Details of these three types of mycorrhizae were already discussed in the previous classes. So you should revise that topic. Okay. So this is about biofertilizer. Thank you.